Gracias. Thank you very much for, I'd like to thank the organizers for inviting me here to give this talk. As was mentioned, the talk is pesticide exposure in occupational or in agricultural settings. I'm with the National Institute for Occupational Safety and Health, which is an agency in the Centers for Disease Control in the United States. I'm based in Cincinnati. We have several offices throughout the country. And just a necessary thing that all of our talks, the things I'm going to talk about and my opinions and the results that I present are my opinions, my position, and not the official position of NIOSH or the CDC. So I'm going to talk about 3 studies that I've done in doing pesticide exposure assessments. The first study is a study done among farmers in Iowa, which is a state in the United States. And it was looking at exposures of pesticides to the farm families themselves. There's something that we call take-home exposure or para-occupational exposure. And it's not fully occupational in the sense that it's not necessarily the wife or the spouse of the farmer and the kids getting exposed from their work, but it's exposure as a result of the worker bringing their exposures home. And this happens in other industries as well. The second study is a study looking at tobacco harvesters. This was done in North Carolina, United States, looking at the amount of pesticides that gets on their hands and how to reduce that. And then lastly, I will talk about a study that I did in El Salvador among some sugarcane harvesters. So the first study was a take-home pesticide exposure. And why study take-home pesticide exposures or take-home exposures? Well, really it's for the families. So as I mentioned, when you do your job, depending on the job and the industry, you may get contaminated with chemicals. So in this case, it's pesticides, and inadvertently bring that into the house through your clothing and your shoes. So it's a worldwide problem. It's health effects and even mortality deaths have been reported in 28 countries. This isn't just pesticides, mind you, but other things like lead and mercury. So if you were at my colleague's talk yesterday, Dr. Grimes talked about some work that NIOSH did in Guyana with artisanal mining and mercury exposure. So that kind of those kind of industries, if there's not good hygiene practices, the workers can get contaminated and bring that stuff into the house. And then with specifically to pesticides, there's been acute poisonings and neurological effects that have been reported with take-home exposures. So in the United States, farmers are the biggest users of pesticides, and therefore are the most highly exposed group. Children may not be highly exposed to pesticides, but because of their developing bodies, because of their size and unique metabolisms, they're thought to be more susceptible to pesticide exposure. And there's been some studies that have shown that parental occupation with involving pesticide application or household use of pesticides have been linked to cancers and other diseases. So how could family members be exposed to pesticides if they're not actually applying pesticides or using pesticides? So there's several routes of exposure. Anyone who's done exposure assessment, industrial hygiene knows the various routes of oral and dermal and inhalation. Well, we can break that down a bit more. So for inhalation exposure for a family member, since they're not actually applying the pesticide, they're not getting the inhalation exposure that way. But if their farm home is close to their fields, there could be spray drift if it's a windy day. The dust in the fields gets contaminated. If it's a dry field and a windy day, the dust can be resuspended. And so they can get some inhalation exposure that way. They could also have dermal exposure just by being outside. You know, kids are in a farm, they're going to go outside, they're going to play in these fields. They may touch the contaminated dust, contaminated soil, other contaminated surfaces. Not necessarily occupationally, but you can get exposure through food. That's one of the bigger ways of getting pesticide exposure for most people outside of occupational settings. And then lastly, especially with respect to small children, is you can have indirect ingestion, which is through hand-to-mouth contact. So children are playing around on a contaminated floor, for example, with dust, and they touch stuff, and then they stick their thumb in their mouth or their fingers in their mouth. And so we call that indirect ingestion. And in the case of take-home exposures, all of these, except for food would be just a normal exposure that most people would get. But the other three, and in particular the hand-to-mouth contact for children, are probably the biggest way of route of exposure. There's been a few studies. Now, these studies are older studies. This was an older talk I gave, but basically this slide is just to show that there's been some studies that have demonstrated that pesticides can be tracked into the house, through various work applications, lawn applications, things like that. This isn't a new phenomenon. It's been demonstrated that if you, for example, apply a pesticide to your garden or lawn, within a few days, you can detect that inside your house. So the objective of this study, this take-home study, was one, to evaluate pesticide contamination in farm homes and the exposure that the farm family members are receiving, and to see if there's any potential environmental, behavioral, occupational practices that may contribute to it and therefore would be helped to reduce it. The study had, in this case, we had 50 homes participate in our study. 25 of them were farm homes. 25 were non-farm homes. To be eligible for a farm home, you had to be using one of our target pesticides. We had 7 pesticides we were analyzing, and I'll have that up in the next slide. The non-farm households, you couldn't be involved in agricultural work, and you couldn't be living on agricultural land. Now, in the case of the non-farm homes, some of them were very close to farm fields. This was a rural area of the United States, so even though you may not have been farming yourself or lived on a farm per se, your home might have been located fairly close to a farm. And we had, in total number of participants, we had 24 farmers, 24 spouses, and 66 children in the farm homes. Similar numbers in the non-farm homes. So I mentioned there were 7 pesticides that we were studying in this study. You can see them up there as atrazine, alachlor, acetochlor, metolachlor, 2,4-D, glyphosate, and chlorpyrifos. All of those are herbicides, with the exception of chlorpyrifos, which is an insecticide. And the names up there, these are the chemical name, the active ingredient name. So the actual name of the product varies country to country. So for example, in the United States, glyphosate is also known as Roundup. So Roundup is the trade name. I don't know what the trade names would be here in Colombia, for example. The same, yeah. So, but just to let you know that these are the active ingredient, the actual chemical name of the product. And the chemical names in brackets, those are actually the metabolites that we measured in. So we did collect urine samples. We're going to go into the methods in a minute. But what you see in the brackets is what we were measuring in the urine. Often when you do urine sampling biological monitoring, it's rare that it's the actual parent compound that you measure, and it's usually some metabolite that's formed. So in our methods, we did both personal and environmental samples. The personal samples consisted of hand wipes of the participants, and then we also collected a first morning void urine sample. In fact, actually, we collected one on the day of our visit. We collected the sample the night that visit, and then the first morning void the next day. And I'll talk a bit more about that. And then the environmental samples, we collected dust samples, hard surface wipe samples. The hard surface wipe samples were both inside the homes of the participants and their vehicle. And we did an air sample and also collected a tap water sample. And then we did a questionnaire interview with the participants to ask about things about pesticide use and household and work practices. And we did sampling on 2 occasions, one sort of early into the growing season, and then another one about 4 weeks later. The range was about 3 to 6 weeks, but on average we're trying to do about 4 weeks apart. And we sampled, what we were sampling in Iowa was corn farmers, so people growing corn, and this was at the start of the study started at the beginning of the growing season, so when the corn was already put in the ground and just starting to emerge, which is when pesticide application tends to happen most often. It's either applied pre-emergence, so before the corn starts to come out of the ground, or just slightly after. Once the corn gets high enough, there's not usually much of a need to apply pesticides anymore because the corn outcompetes the other weeds. And then the only thing you would be monitoring for is insect outbreaks and fungus outbreaks. So for our dust samples, we use this funky vacuum cleaner. This is called a HVS3, or a high volume surface sampler. I don't even know if it's made anymore. But what it does do, it's basically a modified vacuum cleaner that you set a certain flow rate and a certain pressure difference between down here you adjust the height of this, and that makes a pressure difference. And between these two things, the flow rate and this pressure difference, you kind of extract out the bigger particles and just capture the smaller dust particles. And they would be captured through this cyclone here. And you can kind of see this little plastic thing behind this tube. That's where the dust was collected. And we basically had trying to remember, I do believe about a 10-foot by 10-foot template that we'd put down on carpet and vacuum a 10-foot by 10-foot area. For the urine collection, we wanted to collect urine samples from all of our participants. That included women and children, some children who were still in diapers and not necessarily potty trained, so that posed a bit of a challenge. But what we did for the women, if they were so inclined, wanted to, is this, we call it a collection hat, I think is what the term was used. But essentially would sit over the toilet, and a woman could go to the bathroom normally and then pour it into our collection jars. And then these are pediatric urine collection bags that are meant to be put over the genitals of the children. And so we'd have the parents were doing the collection for us, the urine collection. We weren't doing that. And so they were using these and with some mixed success. But in the end, we didn't need a lot of sample. Most of our analysis was done using immunoassay techniques, which really you only need a few milliliters of sample to do your analysis. So it worked out relatively well. For our surface wipe samples, we had a 10 centimeter by 10 centimeter squared area that we would do a wipe on. So we would just put down this plastic template on our surface and do wipes in one direction. With one wipe, use a second wipe and go in the opposite direction. So we do 2 wipes and then stick them into the collection jar. For our air samples, we didn't expect to have a lot of pesticides in the air, just given the nature that this was take-home. It wasn't like during the actual application. So we decided to run a 24-hour air sample, and we had one indoors and outdoors, so that prevented some challenges in the sense that for our outdoor samples, we needed it to be weatherproof. These pumps don't necessarily operate well in the water. And then indoors, we wanted them to be tamper-proof. We didn't want children and family members messing around with it. So we came up with just these basic plastic boxes where we could stick our OVS2 tube. This is a charcoal tube. So it would stick out, and then we could turn our pump on, close the box, and let it run until we came back the next day to pick it up. So in total, we collected over 2,000 samples, several hundred samples each of the dust, surface wipe samples, the hand wipe samples, air samples, a water sample from each home, urine samples. So quite a lot of samples, quite a lot of analysis that had to be done. So it did take a while for us to get all our results back, of course. So the data analysis was done in SAS 9.1, I do believe. We used a mixed effects model. I'm not a statistician. I had a very competent statistician work on this with me. But essentially what a mixed effects model does is it takes into account variables that could be influencing the exposure that aren't necessarily what you're looking at. So for example, the fact that we did 2 visits where we went to a home on one occasion and 4 weeks later, well, there's going to be an autocorrelation going on just because we were at the same home, just difference in time. So this type of model accounts for those kind of correlations that can happen. So the fixed effects, in a mixed effects model, you have your fixed effects. These are things that you put into your model to help predict your outcome. So they included the household type. So was it a farm or non-farm? The visit, was it visit 1, visit 2? We did sampling in different rooms, and I'll show some of those results. So which room that we did the sampling in, the age, and then the sex of the person, and then urinary creatinine. So when you're doing urine sampling, you can get your results in terms of a concentration in urine, but their dehydration or hydration state, I should say, is a factor in what their exposure can be, and there'll be a good demonstration or example of this in my last study on the one in El Salvador. But to adjust for that hydration state, you measure their creatinine in the urine as well, and then adjust your results by that. And you can do it a couple of ways. In this case, we actually put the creatinine results right into the statistical model, but a more simpler way is just to make your results normalized by creatinine. So you would do micrograms per chemical per gram creatinine. And lastly, our non-detect values, to put them into the model, we just replaced them with either the 1/2 of the limit of detection, or in some cases we had values that were reported to us from the lab that were below the limit of detection, and we would use those values if it was lower than 1/2 of that value, if it was lower than limited detection. Now those values aren't very reliable in the sense that you wouldn't want to say that is the value, but it's more reliable than just saying the limited detection, which is just arbitrary. So some next several slides are going to be on the different results. But as you would expect, farm homes had more detectable pesticides in their home than non-farm homes, so not earth-shattering there. What was interesting is the dust samples there's way more detectable pesticide in the dust than any other medium. That is probably a factor of the analytical methodologies as more than anything. And the other potential reason for that is that if you imagine carpet versus a countertop, for example, carpets tend to collect more dust and more dirt and stuff, so there's going to be more residue in something like that, something that's soft surface, as opposed to a hard surface like a countertop. One thing that's really interesting to point out is just here. So in our wipe samples, we didn't have any detectable 2,4-D or glyphosate in any of our wipe samples, despite having 92% of our dust samples having detectable levels of glyphosate and 100% of our dust samples having detectable levels of 2,4-D. This is purely because of the method that we ended up using. At the time, the methods for wipe sampling for those 2 chemicals weren't well developed in terms of the analytical and sampling methods, so our chemists suggested that we use polyurethane foam. So of all the other chemicals, we used a cotton gauze moistened with isopropanol alcohol. And that works really well. The cotton gauze absorbs the alcohol really well. It picks up stuff off surfaces really well. I don't know if you know what polyurethane foam is like. It's also called puff, but it's like a sponge but doesn't absorb much liquid. So when you put isopropanol on it, we would put about 10 mLs of isopropanol on these things. Most of it would drip off. So we'd basically have this dry sponge with hardly any alcohol in it. So it wasn't doing a very good job at removing the residues. And then again, most of our air samples, as we expected, were non-detect. So for all of our pesticides, the dust samples from the farm homes, so not only were there more detectable samples, they also had higher amounts of pesticide in the dust than the non-farm homes. Again, that's to be expected. And then when we did our questionnaire, one of the questions we asked is, one, if they used the pesticide or not. Two, if they did they apply the pesticides themselves, or did they contract it out to someone to apply? They call that a commercial or contract applicator. And so some farmers have these big fields, and instead of doing the applications themselves, they hire companies to do it. So we asked these questions. So in homes, in the farm homes, at least significantly, statistics significantly, atrazine, acetochlor, and metolachlor were higher in the dust where the farmer actually applied the chemical themselves. Now this is kind of what you'd expect, but we didn't see this across the board. Again, it just suggests that the closer you are to using these pesticides, the more likely it is you're going to get it inside your house. One thing that didn't contribute to the exposure was how close the farm homes were to a field. We weren't too granular in our measurement of this. There's a questionnaire again, and really most farm homes are close to their fields. And our what we'd asked them is like whether it was like basically a quarter of a mile, within a quarter of a mile, half mile, or a mile. Now, there's rarely any farm homes that are going to be a mile away from their fields. So we just didn't have the granularity to see this. And the same thing happened with the non-farm homes. They were all tended to be fairly close. So we didn't have a lot of variability in this metric, so we couldn't really see whether there's any difference or not. So to say that distance isn't really a factor, I wouldn't say that from this study. We just didn't measure it properly. And then we asked things on home characteristics such as age of the house, how often did they clean the house, vacuum, whether they were owners, renters, things like that. And none of these factors were related to pesticide exposure. So here's just some slides of some actual numbers of what I just talked about. The numbers in yellow mean that there's a significant difference, statistically significant difference. Just to point out, as you can see, particularly with the dust column here, you got farm and non-farm, and basically almost always it's more detectable samples, with the exception of 2,4-D. Both farm and non-farm had 100% detectable samples. I should point out that glyphosate and 2,4-D are household use pesticides. So the homeowner themselves could go to the hardware store and buy some Roundup to spray on their yard or garden. Same thing with 2,4-D to spray on their lawn. The other chemicals are all agricultural only chemicals. So a regular homeowner can't buy these chemicals. You have to be a pesticide applicator. They're meant to be used on crops and not in your garden and things like that. So it's not surprising to see that we had these high levels of detection in the non-farms for these two pesticides. Now, chlorpyrifos was also detected quite a bit. At the time, chlorpyrifos is just a agricultural chemical now, at least I'm talking about in the U.S. This doesn't necessarily, I'm not sure what the regulatory environment is here in Colombia, but shortly before this study started, chlorpyrifos was, the regulation changed, and prior to the change, you could use chlorpyrifos in and around your home, but there were some concerns about chlorpyrifos having effects with children. So they changed the regulation so that it was an agricultural-only pesticide. So it's possible that some of these high levels of detection are happening because this was just shortly after the study, or shortly before the study started, this pesticide could be used around the home. And then you can just see the wipe and air samples, much less detection. So most of the talk is going to talk about the dust samples, and then I'm going to talk about urine in a minute. This is the dust samples by room. So I mentioned we sampled different rooms. It's a lot of information there, but it's each chemical broken out by the different rooms that we sampled in. So we sampled in the child's bedroom, the changing area, so where the farmer would come in and take off his boots and more clothes. The entrance way to the house, if that was different than the change area. The laundry room, as you'd imagine, that's where the contaminated clothes would get laundered, so we'd assume there'd be some residue there. And then the room that the children most often played in. It usually was a living room, but sometimes there was a separate playroom. And you can see that generally, but not always, it's mostly the entranceway, the laundry room that we're getting the highest amounts, and in the change room as well. So, and these are kind of the three areas where you would expect it. If there is a take-home pattern, the worker comes in with their dirty clothes, changes, goes to the laundry room with it, and then they get laundered. So you wouldn't expect as much in some of the other rooms. The interesting thing again, though, is with glyphosate and 2,4-D, we had 100% detectable samples, so obviously it's the same across the board there. And this is the same slide, same information, but the non-farm homes. And the first thing interesting to see is just that the levels are a little bit less in terms of the amount detected, again, with the exception of glyphosate and 2,4-D, which were pretty ubiquitous. And again, we're seeing it mostly in the entranceway, so the yellow here, here, here. And that would make sense. If you're living in a rural area and there's pesticides being applied around, and you might get some of it on your shoes and things like that, and so that would be the most likely place it would be brought into the home. So this is the concentration of pesticide in dust. So it's nanograms of pesticide per gram of dust. And as I mentioned, we normalized it, or we accounted for the, thinking of something else. So this, we could report this in two ways. So we measured a area on the floor, and so we could report it as in terms of micrograms per area, but in this case we decided to report it as micrograms per gram of dust. So more of a concentration of the dust. And what we're showing in this slide again is all the chemicals broken out by whether they were a farm or not, or whether it was a farm but didn't report using that pesticide or applying it, or whether it was a farm but had that pesticide applied to their fields. And you can see for the most part it's not always consistent, but you go from non-farm to farm without and farm with, and this trend of obviously with farm with having more pesticide exposure. Again, this is kind of what you'd expect to see, but it's always nice to see results that are in line with what you would expect. Again, with glyphosate and 2,4-D, not quite as clear, but again, these are very ubiquitous pesticides in the United States, at least, not just agriculture, but also household use. So let's move on to the urine sampling. So so that was all the environmental samples. We did a lot of urine sampling. Again, some of these results aren't necessarily surprising, but farmers who actually sprayed the pesticide themselves had higher metabolites of that pesticide than if they had the pesticide applied to their crop by someone else, or if they didn't apply it at all. And then there might be an association with time since application, the amount of pesticide applied and acres applied. And I say may or might. It wasn't significant, but there was a trend of that. And that would make sense. The more area you apply, the more pesticide you're going to use, and the closer you measure the urine sample from when they last applied, all those things should result in, you would think, higher pesticide exposures. So this is the fathers in the study, and I'm careful to say spouses of farmers. I'd say 99% of the time in the States, the farmer is the male in the household, but there are female farmers. And in this case of this study, with the exception of maybe one farm, there were all the fathers in the study were the farmers. So these are the fathers who are typically the farmers, and this is their urine results. You can see it's non-farm farms. They didn't spray that pesticide at all. They sprayed it, but someone else did the spraying or the application, and then they applied it themselves. So again, we're seeing especially obvious in some of the chemicals that the farmer, when they sprayed it themselves, it was quite a bit higher than the others. And there wasn't much of a difference between any of the other categories. So the really driver was if they sprayed it themselves. Some other stuff that we looked at, so this was looking at only at atrazine and not the other chemicals, and the reason is that atrazine was our most commonly detected chemical outside of the glyphosate and 2,4-D, and so it was just easier to do some of these statistics on. and we had more samples of atrazine than any of the others. So to do some further analysis, we just looked at atrazine only, and we saw some marginally negative associated levels with the days since spraying. So the further away that they sprayed the pesticide from when we collected the sample, there was a negative association. So the closer you are to spraying, the higher the pesticide exposure. It was marginally positive associated with farm size. So the bigger the farm, the more exposure. Again, typically with bigger farms, you'd use more chemicals. This one here is interesting. It's not what you'd expect, but so farmers in the United States, if they got a lot of money, they have what is called a closed cab, and it has air conditioning. And so when they apply pesticides to corn, they have a tractor, and they pull the applicator behind, and it deposits the pesticide on the field. And so if they've got newer equipment or have the money, they have these nice enclosed cabs that are air conditioned. Some farmers use a cab that doesn't have air conditioning, so they just have the windows open and stuff when they're applying. And you would think that there would be more exposure in these open cabs, but we for some reason found that it wasn't statistically significant, but they tended to be higher in these closed cabs, and we're not quite sure why, but it could be just these cabs get contaminated over time and they're not routinely cleaned. And then the urine levels were not correlated with these, with the amount sprayed or the acres, the area sprayed or the use of PPE. Again, that's probably more of an artifact of the questionnaire and how it was developed than actual results. So that was atrazine. For 2,4-D, we were able to just do this analysis as well. We had pretty much the same results as we did with atrazine across the board, just different sort of values. But then, and again, this closed cab, not quite sure why. And then the levels weren't associated with at least farm size and PPE, but a little bit associated with acres sprayed. So that was one difference. Let's move on to the families now, the wives and the spouses and the children in the study group. Again, not unexpected, but farm children and their spouse and the farm spouses had higher exposures than the non-farm children and spouses, but it was only significantly different for atrazine. And then, in particular, when atrazine and chlorpyrifos were applied by the father, the children had higher exposures as well. And there tended to be a trend of decreasing exposure with the age of the child, which you would sort of expect as well. As a child gets older, they're playing less on the ground and crawling around and putting things in their mouth that they shouldn't, and maybe even playing less out in the fields and stuff like that. Getting more on their iPads and iPods and playing games as opposed to going outside. So here's just some values. It's a bit of a busy slide, but just this is the geometric mean between the non-farm farms. So we have atrazine, mother, child, chlorpyrifos, mother, child, metolachlor, and glyphosate. So with the mother and the child, we didn't look at all 7 pesticides. We just looked at just these ones, partly because of the analytical methods we were using. For the urine, we were using immunoassay, and for these samples, and we didn't have analytical methods for the other pesticides. And you can see that for atrazine, there was a significant difference between the farms and non-farms, but really there's a trend of higher from farm to non-farm on all of them, with the exception of glyphosate. And then this is looking at the urinary concentrations for mothers and child and application status, so whether the pesticide was applied to their farm or not, and if the farmer applied it or a commercial applicator. And you can see, again, with the father application, it's typically higher, but it's not a big difference here in atrazine, but in chlorpyrifos, there was no custom application, but the father applying is a little bit higher, and it's the same with the others as well. So typically, if the father is actually applying the pesticide themselves, there's going to be more potential exposure in the families. We saw significant associations between the urinary levels and the actual amount we found in the dust, but only in the non-farm homes. We didn't see that same significant association in the farm homes for some reason. And it could be that that's really, there's other drivers of exposure in the farm homes. And there was no association with tap water pesticide exposure. In fact, I don't think there was much in the water itself. There was no associations with farm size, the numbers of acres that were applied, the amount applied, days since last application, whether the kids were involved in farm chores or whether they were playing in the fields themselves. So those things were, these were things that we got out of the questionnaire, so it's all self-reported, so there could be some of that playing in it, but none of there was no significant associations seen with those factors. And then the father's urinary metabolites, obviously in the farms, the father's metabolites correlated much better with the family's metabolites as well. So just looking at an association between the dust and the urine, how well they were correlated. They were relatively fairly well correlated, and in some cases significant. So the darker ones, these bolded, are significant differences. Or there are significant correlations, I should say. So we did see some correlation between what was found in the dust and not, and between the dust and between the urine samples. I want to move on. So I'm going to take these urine samples and talk a bit about dose. So far I've just been talking about exposure, and exposure doesn't necessarily mean disease, and it doesn't even necessarily mean dose, right? It's what gets inside you is the issue. So in a way to get at dose, without knowing exactly what's going to, what is the actual dose of these, in terms of measuring it specifically, we did a calculation where we factored in things such as their excretion rate and their body surface area and things like that. So this urine samples was a spot sample. So if I really wanted to get dose, I'd have to collect urine samples over a full 24-hour period if I wanted to get a daily dose. So we only collected one sample, that's a spot sample. But we can take that and extrapolate it into a dose, and that's what this calculation is about, without going into it too much, but it's basically taking their surface area and the excretion rate and the metabolism of the pesticide itself and coming up with a dose using this calculation. And what we ultimately get is an average daily dose, and we can use that value to compare to threshold values. So in the United States, the Environmental Protection Agency, or EPA, sets what they call reference doses, and we can compare this value to the reference dose, and the reference dose is supposed to be essentially a safe dose. So before we talk about the reference doses, just to show you the geometric mean doses for farm children were higher than the non-farm children, except for glyphosate, which was pretty even. Again, this is going to essentially follow the exposure, because I'm using the exposure values to extrapolate a dose, so it shouldn't be different, and it isn't. And you can see the highest dose estimates are here. So the unit for a dose in this case is micrograms of pesticide per kilogram body weight per day, and that's a very standard unit that's used in pesticide regulatory decisions in Canada and the United States. And this is the farm children levels, and just on the surface you can see that the farm children had higher doses than the non-farm children. No child had an overall dose estimate that exceeded the chronic reference doses. So the EPA publishes acute reference doses and a chronic doses. So acute would be something more like a high exposure event. But then typically what happens is you don't have these high exposure events, you have low level exposure over time, which is a chronic exposure. And the EPA puts out both, usually, not always, but usually an acute and a chronic reference dose. But in the case of the chronic reference doses, the values didn't exceed it for atrazine, metolachlor, and glyphosate, but they did exceed every one of them for chlorpyrifos, for at least the population-adjusted doses, and I'll talk about that in a minute. And again, there was a trend of decreasing dose with increasing age, but we did see that with exposure. So, and the doses were similar whether you were male or female child. So here's the values. This is our geometric mean doses. and with the significance level here. So atrazine was the only one significantly different, but all of them were higher for the farm versus the non-farm, just not always significantly. This one's marginally significant, but these two weren't significantly different. And then to look at the reference dose, so these are the EPA reference doses. We have an acute reference dose and a chronic one. With the exception of metolachlor and glyphosate, there isn't an acute one, but just a chronic. And this is the percent of children with estimated doses exceeding these various reference doses. So let me just talk a bit about what a reference dose is and how they're calculated. So they're based on toxicological studies, usually on rats. And what they do is there's various effects. The most common is usually a body weight decrease in the rat, and they call that they call that a NOEL or NOAEL, which is a no observable adverse effect level. So they dose the rats at various levels, and they pick the dose where they don't see any effect. And if there's a body weight change, well, that sometimes is an effect. So they're usually pretty low as it is in terms of the amount of pesticide. But then what they do is add a safety factor in to get their reference doses. So usually to account for differences between species, so going from a rat to a human, they add a 10-fold safety factor in, and then usually to account for variability within humans, because we all metabolize and react to chemicals differently, there's another 10-fold. So usually the actual reference dose is about 100 times less than the no adverse effect level. So you're already getting down pretty low, so there's all this safety factor in. Now, in some cases, they'll come up with the EPA will come up with something called a chronic population-adjusted reference dose. So what they do then is if they think there's segments of the population that are susceptible to a chemical, they'll add an extra 10-fold safety factor. And so now you're talking about 1,000 times below the actual no observed effect level. And so in the case of chlorpyrifos, they came up with a population-adjusted dose, and same with atrazine, where they added this extra safety factor. So you can imagine these are basically factors as you go down, like 100 times less than the actual what you would see as an adverse effect in a rat, or 1,000 times less. So a NOEL then, we didn't see any of our doses that would exceed the NOEL, which is good, because if they're exceeding the NOEL, then they're possibly in a toxic state. Nothing exceeded the acute reference doses, and the acute doses are usually a lot higher. But now we start to see some in the chronic, and in particular with chlorpyrifos, most of them, and then with the chronic population-adjusted dose, every child exceeded that. And even the acute, they're starting to exceed. So the values, the levels we saw in children, so the rest of the pesticides, you can say there's exposure, but if you trust the regulatory agency, you can say that exposure is well within the normal limits and a safe limit. But with chlorpyrifos, that's not what we're seeing. So either they're being highly exposed, they are being highly exposed, but there is a possible explanation, because these children aren't necessarily getting sick. But the metabolite that we use to measure chlorpyrifos in urine is called TCP. And that metabolite, they've since found that you could take a food sample, for example, and that might have chlorpyrifos in it, and it can be broken down in the food, and so you can actually ingest TCP directly, which is in and of itself isn't really toxic. It's the parent chemical and the steps that get to TCP that causes toxicity. So some of this high level of chlorpyrifos that we're seeing in the urine and the dose could be related to actual ingestion or exposure to TCP directly, and that's not something we measured for in this study. In fact, I wasn't aware of this until after the study came out, and I saw then some other studies that were showing that TCP itself can confound what you're seeing. So all that is to say is definitely the kids were getting some exposure to TCP. We're not 100% sure it was all from chlorpyrifos. So just some conclusions on this study, and then I'll get into a couple other studies quickly. But just as I mentioned, the farm homes seem to be more contaminated than the non-farm homes. The dust was a better sample matrix than, say, the wipe samples or air samples. Farmers tended to have greater exposures when applying it themselves versus having someone else apply it. All these things are kind of intuitive. Children and spouses had higher exposures. The urinary metabolites were more correlated, or the farthest metabolites were more correlated with the farm family members. And the pesticide levels were higher in children when the father applied the pesticide. So that it's also suggesting that there is a take-home pathway, that some of this exposure at least is coming from the father's occupation or the farmer's occupation. A bit about the dose estimates. As I mentioned, all the chlorpyrifos dose estimates were higher than reference doses, but I kind of explained why that may be. And certain things that we thought might have some limitations that I should say about the study is relatively, I mean, it's still 100 people in the study, but it's still not a large study. And there's large exposure variance. So you need to always interpret, exposure data has to be interpreted with caution always, just because of some of these issues. So let's move on to tobacco harvesting. So this study was also done in the United States and North Carolina. When they harvest tobacco in the United States, they have these crews, at least at this farm anyways, it does vary depending on the region, but here they have these vehicles with crews of 6 people, and here's a better picture up here. But what you do is you get 2 people sitting here, there's 2 on the other side, and this machine goes through the rows of tobacco, and these guys pick it off the tobacco, off the plant itself, and they stick it into a conveyor belt, and I have a picture of that, and it moves up to here, and then there's 2 guys here that grab it out of here and throw it into this bin. So there's a lot of contact with the crop in this case. So we're looking at direct occupational exposure here, a lot of contact with the plant, and there's usually pesticide residues on plants. And this is just a picture of them washing their hands after. So the reason why we did this study is in the United States we have something called the Worker Protection Standard. So you probably know about OSHA, and they have their standards. Well, one thing that OSHA doesn't regulate is the agricultural industry, and so that is in the domain of the Environmental Protection Agency. So they have something called Worker Protection Standards. And one of the things in the standard is that when you have workers in the field, they have to have access to facilities to clean their hands and stuff. Now, it isn't very well defined, and it could be better now than when I last looked, but they may have their, if they're out in the field, and these fields are acres and acres of crop, it might be a 15-minute walk back to go where they have to go wash their hands, or even 20 minutes. So they're not always washing their hands, and what's better is having something right there on the tractor with them. But what we wanted to see is, how much exposure these workers were getting, and how much exposure these workers were getting, and how much was on their hands, and whether washing with soap and water actually helped. And hands, when it comes to pesticide exposure, or at least in occupational agricultural settings, dermal is the largest route of exposure. It counts for way more than inhalation, roughly around 80%. And in the dermal realm, it's mostly on your hands. A lot of that is on your hands. So this is a picture of the workers that actually pick the tobacco off. So you can see they're totally enveloped in this tobacco, and they throw it into these conveyor belts that pull it up to the top. And these are the guys at the top that grab it out of here and put it into the hopper in the back. So as I mentioned, we wanted to, the chemical that we were studying in this one is acephate, which is another herbicide, and it's applied to tobacco. And will soap and water be effective? One thing I should point out: acephate is a very lipophilic compound. So it absorbs water. It doesn't repel water, so it's a good candidate for being removed with just using soap and water. There are other chemicals that water doesn't work very well on, but this isn't one of those. And this farm, we just went to one farm in North Carolina. North Carolina is a big tobacco producing state, along with Kentucky. And we had 12 harvesters, 2 teams of 6, so each 6 people would work on one of these machines, and all Hispanic males. 5 fields were harvested, and they would collect the bottom few leaves from the tobacco plant. And as I mentioned, 4 of them working on the bottom with 2 on top. And we collected hand wipe samples from each of them. Total 96 total we collected. We would collect them, so they would start their day, then we would collect our first sample at their noon break, their lunch break, and then another one at the end of the workday. So we collected on 2 occasions.
How do you do hand wipes? Because typically when you do surface, you have a specific area.
Yeah.
What type of
I'll describe that in a minute. Okay. So the question was, how do we do the hand wipes? And so I'm going to describe that in a bit. In addition to doing the hand wipes, we also collected wipes on the leaves. So there are these big leaves, so we could do wipes on that and see what pesticide residues were actually on the leaves. So to do the hand wipes, what we did is we took 4-inch by 4-inch gauze, cotton gauze wipes that we moistened with isopropyl alcohol. And we would take we'd use 2 wipes per hand, and we'd take 1 wipe and just wipe all over the hand, stick that in a jar, and then take a second wipe and do the exact same thing. So two wipes back to back. That was our 1 sample with these 2 wipes. And in the question that was asked was also, how do you standardize that? Because when you do a surface wipe, you have a standard area. Well, for hands, what we would do is the EPA has a guide that they publish for various factors. It's called the Exposures Factors Handbook, and you can find it on their website. But they have these standard rates for surface area of bodies, so different parts of the body. So if you want to do dose calculations or things like that, or you can factor in things like, they have like a standard height, standard weight, standard surface area. So if you don't know these things on the people you're doing your sampling on, you can just do these standardized measurements. And then what we did is we randomly did the left or right hand so that we weren't, there may be a handedness in this type of work, and so maybe one hand would be more contaminated. So we didn't always want to sample just the left hand in case that was the problem. So we would just randomly do the left or right hand. And then if we picked the left hand, so what we did is we did a wipe before they washed their hands and then right after. So if we did their left hand, we'd wipe that, then wash their hands, we would sample the right hand after. And the reason is presumably if our wipe sampling method's working well, we're already removing a lot of the pesticide before they've even washed their hands. So you want to make sure you're not getting an artifact of that sampling method. This is just a picture of how it's done. This isn't the actual people in the study, but it's the way the sampling is done. So the field workers would go and put latex gloves on and take their moistened isopropyl moistened gauze and wipe each hand and put it in the jar. And then we would change the gloves between samples so that there wasn't cross-contamination. For the leaves, you saw the tobacco leaves are pretty big, so we used a 10 centimeter by 20 centimeter area template that we would clip to the leaf, and then we would do our wipe sample within that area, so we'd always have a standard area that we collected from the leaves. And we would do 2 leaves per plant and combine them into 1 sample. And we sampled 3 plants per field, with the exception of 1 large field that we did 6 plants. And so we did 4 of the 5 fields that actually they were harvesting from. We sampled from 4 of those. We did our analysis using GCMS. We used this was some older study work, so it was an older SAS version. And again, we use this mixed model procedure that I talked about before because it accounts for any correlations that could be happening. And so here's our results. The average acephate level was 1.4 nanograms per centimeter squared. So this is on the leaf itself. So there is residue on the leaves. And then we saw a lot of this was being transferred to the hands. And washing was very effective, and I'll show you some of the actual results. But when you have a compound that is dissolved well with water and you use soap and water, it's going to remove a lot, and that's what happened in this case. So up to 96% of the residues were removed just by washing. So it does go show that washing your hands is a good thing. And there was some accumulation over time, so you'd remove it, but not all of it. And you did see a little bit of a trend of that. I mean, we did only 2 days of sampling, so it was hard to really see that trend, but there was some hint of it. The wipes on the amount on your hands was positively correlated with the amount on the leaf. So obviously, the more on the leaf, the more there was on your hand, and this was significant. There wasn't any difference between whether it was a left hand or right hand, so there wasn't really a handedness factor in this type of work. The job had, so I talked about the 2 jobs. There was one person or people on in the tobacco itself, and then the people at top. So there was a difference between what job you did on your pre-wash levels. After the wash, there was no difference, but that's presumably because you're removing most of the pesticide. So it was better to be on the top than on the bottom. And here's some of the results that we saw. So this is this is the leaf samples, so what we actually collected on the leaves of our 5 fields and the number of samples that we collected. And this is just the arithmetic mean. So we are seeing some, and we didn't sample the 5th field, so that's why it's not applicable. But you can see that we had small amounts on the leaves. And then you can see the samples. So we did our day 1 and day 2 in the morning, the afternoon, pre-wash, post-wash, and big differences between pre-wash, post-wash at all our sampling periods. So it's just. This is the same data, but instead of looking at it by time, it's looking at it by field, just to see if there's any difference, whether it mattered what field, but having all the samples from each field. And again, you see this big difference between pre and post-wash. This is just a visual. Sometimes it's easier to see this stuff in graphs, but it's very clear. Pre-wash samples, post-wash samples. And then over time, this is where you can see there's this little upward trend even after washing your hands. So the second day, the second sampling period, even though they wash their hands, they're getting more samples. So there is this accumulative. Even though you are washing, there's still stuff left on the hands, and we're seeing more over time. Looking at some things like which hand or which sampled or which job you did. As I mentioned, no difference between the right or left hand in pre-wash or post-wash samples. I mean, there was a difference, but just not significant. But in the job, not in the post-wash, there was no significant difference, but it was marginally significant. You had more exposure if you worked on the bottom of that machine as opposed to the top. And then we, again, a test for trend. You can see that there's definitely post-wash, there was a significant trend of increasing exposure. And almost, even with pre-wash, there was a significant trend, just not as significant. This slide, we didn't include the leaf wipe values in our statistical model. So knowing that your hand exposure is correlated to your leaf exposure, if you exclude the leaf exposure, you're not accounting for that, and that there's this correlation. So it could be that these results were just because of what was on the leaves. But in fact, if we ran the same model and then included the leaf values in it, and our differences get even bigger. So it had nothing to do with what field they were in and everything to do with what job and over time. So just conclusions, a bunch of the acephate is getting transferred to their hands in this case, and because of the type of chemical that we were measuring, it was very amenable to being removed with soap and water, so washing hands was very effective, but not all of it was removed. So last study to talk about is this study we did in El Salvador in the sugarcane industry, looking specifically at the cutters, the people who go and cut the cane. And just let me tell you, I've seen a lot of awful jobs in my professional career. I don't just do pesticides, but cutting sugarcane in the heat of El Salvador is one I never want to do. So thank yourselves for your education. So the reason why we're doing this study in El Salvador, we were asked to go down there by PAHO to help. I'm not sure how many of you are aware of this, but there is a a lot of young males, particularly in Central America, but other hot environments like Sri Lanka, India, Thailand, are getting chronic kidney disease, and they call it chronic kidney disease of unknown origin, or CKDU. Sometimes it's called non-traditional, or CKDNT. And it's also in Central America called Mesoamerican nephropathy. And the reason why they call it this is typically chronic kidney disease as a result of high blood pressure and diabetes. And these are, and typically older people. In this case, these are young men working in hard labor industries outside that are getting this disease, and there's no obvious reason for it. And so some of the thought is that it's, this is probably most of the case, but that it's related to heat stress and heavy workload and dehydration. But then some people think that there's maybe cofactors involved. So, toxicant exposures, using anti-inflammatory painkillers, maybe exposures to some infectious diseases, alcohol abuse, things like that. And the reason why it's happening largely in hot environments, but it's not happening consistently, so not all jobs are getting it. So a construction worker, for example, in these areas aren't there. I mean, you may see it in some of them, but not nearly to the extent you see it in sugarcane workers in particular. And so there's thought that pesticides might be playing a role and that it's a cofactor. And so NIOSH, given our expertise in this area of doing pesticide exposure assessment, NIOSH and myself in particular was asked to come down and do a small study to see if there was any pesticide exposure. So we were looking at that aspect of it in terms of chronic kidney disease. But these sugarcane workers who are getting chronic kidney disease, I mean, they are working in 35 degree Celsius weather with high humidity, 6 hours a day, nonstop. They get paid per piece, so they don't get paid by the hour. They get paid by the amount of cane they cut. So there's not an incentive to stop and rest. So they work hard until their shift is over. And unlike in the U.S., down there the rules are a little different, and so they might bring their own water. So maybe they have a liter or maybe 2 liter jug of water, but when it's out, they don't have any more water. So there is an effort to try to improve the conditions that they get. If they were the working conditions, if that was happening in the United States, it would be mandatory that they have a rest break every hour in shade or in cool environment and be given water at the field. So trying to implement those kind of standards is one of the things that people are trying to do down in these sugarcane areas. But we were asked to look at the pesticide exposure of it. And pesticides, they often become the boogeyman, and rightly so. I mean, they're designed to kill, right? They're designed to kill a plant or kill an insect or kill a rodent. So exposure to them, they're toxic chemicals, but the regulatory agencies try to make them as safe as possible. But El Salvador, because of this problem with chronic kidney disease in sugarcane, was proposing to ban 53 pesticides. Now, I'm not a pesticide advocate, I'm far from it, but if you ban 53 pesticides that are registered to be used on your crops, I mean, that's just going to be a huge disruption to the industry. And they were proposing this ban without having any evidence to suggest that this was the problem. So we went down to do a small study. Just a bit about the harvesting process, if you're not familiar with sugarcane. At least in El Salvador, the harvest typically runs from November to March or April. It's done manually, although it's being more and more mechanized. In the United States and in Australia and other cane-growing regions, they use machines to harvest, but in Central America, at least, it's still done mostly by hand. And glyphosate, or Roundup, is applied to sugarcane. Interestingly, it's not applied as a herbicide, because glyphosate generally kills anything that's green, unless you buy Monsanto seed, which is glyphosate resistant. But sugarcane, what they use it for is it stops the plant from growing, and it concentrates the sugars into the cane. So about a month or so before they harvest, they'll apply this glyphosate to ripen, basically quicken up the ripening of the cane and get more sugar in it. And then a day or two before you harvest it, they burn the cane. So this is a picture of the cane being burnt. The reason for that is it makes it easier to harvest. It doesn't affect the cane stalk at all, which is where the sugar is, but it gets rid of all the leaves and stuff, and the leaves have sharp, prickly things in them, so that's not fun to cut green cane. And then in the case of places like El Salvador and stuff, it's to also get rid of the snakes and rats and stuff that are living in the cane, so the workers don't have to deal with that. So some of the recent research that is suggesting that pesticides could be a possibility. There have been some studies that have suggested pesticides are a cause, but all of these studies, none of them actually measured exposure to pesticides, so it was self-reported pesticide use, things like that nature, which was with the exposure measure. So it's not the best way to say, it's not the best exposure metric is by just reporting it. So we went and did a study. We had 40 sugarcane cutters in our study. We've had 20 in 2 locations. So there is some difference in terms of the amount of chronic kidney disease that's occurring. It varies by location in El Salvador, at least. I don't know if this is the same at other countries, but they see more of it in the coastal regions than they do inland. And the reason they think is the coastal regions are warmer. Inland, there's more elevation, so it's a little bit cooler. But we decided to sample in both locations just for that reason. And we did sampling over 3 days, and we collected an air sample, an area air sample. So this is a pump with a basket with pumps. I'll have a better picture in a minute. We also did hand wipe samples, same way we did it in the other 2 studies. And we also collected urine and water samples. And in this case, we did sample analysis for glyphosate and 2,4-D. I always thought this was a neat picture coming from the United States. So being a government worker, the United States State Department had requirements. And, El Salvador, there's a lot of gang activity and criminal activity, and we were leaving the main city to go out to these rural fields. Some areas are controlled by gangs, so it was mandated by our State Department for us to do our work. We had to have armed guards with us. So it was a bit weird. I don't usually see guns often. I know the U.S. has a lot of them, but I've never touched a gun in my life. So to be that close to one was kind of odd. So here's just a picture of doing some hand wipe sampling. For anyone who's done exposure assessments out in the field, it's challenging. There's a lot of logistics to get over and hurdles. It's doubly so when you do it in a foreign country, and then even doubly so when it's a foreign country that doesn't have necessarily the resources you're used to. But we made do, and we had some tents set up because it was hot and there was sun beating on us. And we would collect our samples in the field and stick them in coolers and try to keep them cold. We collected urine samples as well. The sugarcane cutters start very early, about 4:30 in the morning, so it's still dark. They want to get a lot of the cutting done before the heat comes. Usually by 9 a.m. it's 35 degrees and humid. So we would collect a sample from at the beginning of their shift and then at the end, which was around noon. So this is just a picture of us getting our samples ready. And then here's the picture, a better picture of the sampling for the air sampling. This was a bit of a challenge. We didn't want to but the ideal way would be to put a pump on the worker themselves and have the sampling tube close to their mouth. But it's already hard work. It's already hot. So we didn't want to burden them. We had 3 pumps in here, and to put 3 pumps on them in this very physical type work just didn't seem right. So we tried to make do with doing area samples and keeping it as close as we could. And they moved pretty fast. So every 15 to 30 minutes, we'd move the pumps along and try to get them as close as we could, but it was not ideal in terms of collecting air samples, and you'll see that from the results. And just a note, I found this interesting, but if the cane field is close to where they live, their families will come out and have breakfast with them. So this was probably around 8 or so in the morning. They've been cutting already for a few hours, but the families come out. So kind of takes you back to this take-home exposure. So when the worker, they have these exposures, but depending on the environment, the families might get some exposure as well. Now, we didn't include families in this study, but I just thought that was interesting to see that the wife and son are out there having breakfast in the field with their family member. And I mentioned this is hard, hot labor. They are, and it's men and women that do it, and they are, you take the cane, take a bunch of them in one hand, and you take a machete and you hack it at the bottom, at the base of the cane, and then you've got to hack off the leaves off the top, and then you cut it into these shorter sections and pile it up for a machine to come in later to load them onto a truck. So it's just hard physical labor. In fact, there's been some studies on the amount of workload that's produced out of this, and it's equivalent to running a half marathon. And so they're doing this every day, running a half marathon every day for about 4 or 5 months. They usually get 1 day off a week. Usually Sunday they take off, but not always, depending on what needs to be cut. So for our results, we didn't see any, all of our samples were non-detect, and I mean, I do think that was partly because of the sample method. The pumps just weren't close enough to the actual cutting itself, but when you do watch the cutting, there's a lot of dust that's getting kicked up and stuff, so if you had a pump near the base of the cane that was being cut, I'm sure you would pick up something, but we just weren't close enough. And we even measured not just pesticides, but dust, and we weren't seeing any dust on these filters either. So our hand wipe samples were also non-detect. That I found very surprising. Their hands were very dirty. Did you have a question? Why? Just as a measure of the dust, like these are very dusty fields. There's a lot of ash from the burn. So the question was, why did we measure respirable dust? There's a lot of dust and ash. When you watch them, there's a cloud of dust around their feet, and there's some thought that there might be pesticides in this dust and stuff. So as a sort of a surrogate for pesticide exposure and other exposures, I didn't talk about it here, but silica is also thought to maybe be an issue, and so we thought if we could measure silica. And I was going to do silica analysis, but then when all the dust samples came back non-detect, I said, don't bother wasting my money on the silica because we had no dust. So that was the reason. But what was surprising is our hand wipe samples were all non-detect, and their hands are very dirty. They're all black. This cane is all, it's burnt, right? So it's soot. It's like walking through a fire pit. So that to me suggests that there isn't really a lot of occupational exposure going on, just that result alone. But we did find samples in the urine that were detected positive for pesticides, but not a lot, only about 20% overall. So for glyphosate, 20% of the samples, 21, and 2,4-D, 17% of the samples. So if they were being occupationally exposed, we would have seen it on their hands and we would see it in their urine more than this. Now they're probably getting some exposure, maybe from this, but more from maybe other sources. These workers are all very poor, so they do a lot of subsistence farming on their own to supplement, either their income or just to have food sources. So there could be some home use of these things. They live near fields and stuff like that too. So ideally, it'd be nice to measure these people over a year and see if there's any sources of exposure that could be outside of occupation. One thing to point out, if you look at the AM versus the PM, AM versus PM, we had way more detectable samples in the afternoon versus the morning sample. So the morning sample was pre-shift before they started working and then after. So that might indicate that they are getting exposure throughout the day. At least that would suggest that. And when you look at the actual amounts, so this is unadjusted, so this doesn't take into account for creatinine, you do see this AM versus PM higher trend. Now, this is where correcting for hydration really matters. In the bottom slide, so this is 2,4-D, the bottom slide, you actually see almost a reverse trend between the averages. And in this case, now we're adjusting for the amount of creatinine that's in their urine. The more dehydrated they are, the more creatinine you see. And so really what this is showing is the reason why we got these higher numbers in the afternoon when we didn't adjust is it's just more concentrated because there's less urine, there's less volume being excreted. Same amount of pesticide, but less of a volume. So really what's happening isn't necessarily that they're getting more pesticide exposure over time when you, they're just getting more dehydrated. So that's just a plug for if you're ever doing biological sampling, urine sampling for pesticides, or any chemical for that matter, you should measure creatinine in the urine because that indicates hydration and can affect your results. And that's 2,4-D. This is glyphosate. It's the exact same picture that you're seeing with the glyphosate. And so just to wrap up, pesticide exposures for sugarcane harvesting, at least this is a small study done only on a very short period late in the harvest, but it seemed to be minimal. However, there was some exposure coming from somewhere, and we did see a little bit in the urine. However, probably not from cutting cane, at least in this case. So really more pesticide exposure work needs to be done in this for this issue of CKD. Not to say that pesticides is the problem, but to rule it out. Really, it's this hot environment, hard work issue. But a lot of people still say it's pesticides. And in fact, you talk to these workers and their families, they say, no, it's all pesticides, it's all pesticide. So more work needs to be done on this, just to say that, you either say it is or it isn't, but in my opinion, I don't think it is the problem. And, we should be spending our resources elsewhere. And just want to quickly acknowledge all these 3 studies. There were several people involved in these studies that helped out in various ways, from helping with the analysis to doing the analytical analysis, the statistics, the actual work in the field, recruiting people. So just a ton of people involved in doing these studies, as you know. And with that, I will take any questions. We got 5 minutes for them. No questions? I'm sure there's a language issue. You speak English or Spanish? Maybe someone can translate? Can someone translate? Oh, yeah.
Gracias. En el primer estudio, aunque no lo mencionan al final y no aparecía como los objetivos del estudio, ¿pensaron o hicieron en algún momento correlación entre los niveles de TSP y niveles de acetilcolinesterasa en los niños que tuvieron chronic PAD importantes, o los resultados importantes del chronic PAD?
I'm not sure I entirely understood the question, but it had to do with TCP and the children and how it correlated. I mean, I do believe with the TCP, as I mentioned, I don't think their results are coming entirely from pesticide exposure, from chlorpyrifos exposure. I do think some of it is, but as I mentioned, you can take a salad, at least in the United States, you take a salad and you send it off to the lab. They can measure for chlorpyrifos and measure for TCP, which is the metabolite of chlorpyrifos, and they will find TCP in that salad, and maybe or maybe not chlorpyrifos. So it was hard to tell because we didn't know that this was an issue, that TCP is something they can be exposed to directly. It was always thought that TCP was only metabolized in the person. And so if you measure TCP in the urine, therefore you're seeing chlorpyrifos exposure. But that isn't, we now know that's not entirely the case. So there may be, if I were to do this again, there might be different metabolites that I could measure that are more specific to chlorpyrifos from pesticide exposure, or I would try to get an idea of how much TCP they're exposed to outside of the exposure. So if I collected a dust sample, for example, I would collect it and have it analyzed for both chlorpyrifos and TCP and help use that as background to eliminate some of the TCP that I see in the urine. Did that answer your question? I'm not sure if I understood your question well, so sorry.
In the second presentation, you mentioned significant exposure.
Mm-hmm.
But it was nanogram. So
yeah.
Nanogram is significant exposure when you're talking about it?
So the question was, in the second study on tobacco harvesting, I mentioned significant exposures, but the exposures were nanogram levels. When I said significant, I'm talking statistic significance. So statistically different, significantly different from one thing to the other. Nanogram exposure, I don't know if that's significant or not in terms of your health. It's a low level of exposure in some cases. So if you really want to know if that means something for your health, you have to compare it to reference doses like I did in the first study. And I didn't do that with this study. But so I don't know, that's kind of a caveat with exposure assessment studies is you find out what exposure you can. Nowadays, the chemical methods, the analytical methods are getting really good. You can measure really small amounts of a lot of chemicals, but what does that mean? Is it really a health problem or not? I mean, we're all exposed to things, just at what level? And it's the amount that really is a matter. So I typically don't do that side of it. I'm just measuring exposure and seeing what's there. And it's a good example in the last study where you're not seeing anything at all. Like they're saying pesticides are causing chronic kidney disease, or a factor at least. But if we're seeing nothing or very low amounts, is it really the problem? So, but yeah.
I know it was not part of the presentation today, but the biggest concern with pesticides is more the application. Here it was more the people after application.
Yeah. So the question was, is maybe the bigger concern for pesticide exposure amongst the applicators, and what I showed was a lot of post-application exposures. Yes, that is the case. The applicators usually have more exposure than the non-applicators or post-applicators, but we were just interested in seeing these exposures post-application to see what levels they had. There's been a lot of studies with applicators, not as much with the post-application. And in the case with the last study, the sugarcane harvesters, they're the ones that are getting the chronic kidney disease. The pesticide applicators in sugarcane industry aren't getting chronic kidney disease. So if people are saying it's pesticides, I mean, I did have one person at another talk say, well, why, if you're measuring pesticides in sugarcane, why are you looking at the cutters? Go to the applicators. They're going to be more exposed. And I would say, yes, they are, but they're not the ones having the problem. So this pesticides issue keeps coming up, and it's a potential issue outside of the application as well. And so that's why we looked at post-application. Yes.
Bueno, es evidente que en la agricultura aún hay mucho vacío por el cuidado y la seguridad y la salud del trabajo, pues, de las personas. ¿Qué podríamos hacer para mejorar esto?
So, yeah, there is a gap in care in agriculture, and I would say that gap even exists in the United States as well. So this is the last question because I'm getting the red sign saying I got to stop. But I'll answer your question at least. So what can we do to improve that? It really comes down to regulation. So, in the United States, agriculture is regulated by the EPA. The EPA as a regulatory agency I don't think is quite as strong as, say, OSHA. And so the OSHA standards tend to be more enforced. Now, I don't know about Colombia, for example, or in the case of El Salvador, but the regulations are just not there. So the first thing is regulations, and that requires strong governments, stable governments, and that's not always the case in some countries. Outside of that is just good practices. And some companies are very good at that and want to do what's right for their workers. So in El Salvador, the company we went to, one of the reasons why we went to them were we have to get permission and be allowed to go on these company property. But they wanted to help their workers. They wanted to find the answers to these problems. So I think, having regulations and then just educating and having decent employers that, realize that, you need your workers to keep your company going, and they're a very valuable asset, and they're often not treated that way. Especially these hard labor jobs that don't require education and skill. Anybody can do it, and so they just don't care. Someone quits or gets hurt or dies, they'll just find someone else. So. All right. Well, thank you very much, everyone.